Okay, welcome to chapter 5. This chapter is called The Working Cell. So we're going to start with the plasma membrane and its function. So remember, the plasma membrane is the membrane that surrounds every cell, and it has several roles. One of its main roles is to separate the internal and external environment, the internal environment of the cell, separated from the external or the extracellular matrix, therefore allowing a cell to have its own internal biochemistry. Okay, it's also the gate and gatekeeper, which means of the cell, of the factory. We use the factory analogy in chapter 4. Therefore, it also regulates which molecules can come in and which molecules can leave, and also it determines how fast they can leave or enter, or whether or not they can enter at all. Okay, it's made completely out of, well, primarily out of a phospholipid bilayer, and it has proteins that are embedded into the plasma membrane. All right. So let's take a look at what that looks like. Okay, this is the fluid mosaic model. Okay, let's break down this word. Fluid means not solid. Okay, mosaic is in reference to a piece of art, or mosaic art, which is a collaboration of different tactiles in different colors. Okay, and then model is just simply means model. So they call this the fluid mosaic model because the plasma membrane is indeed a mosaic of different components. So let's start with the first one, the phospholipid bilayer. So this is the phospholipid right here. We're going to zoom in on it. Okay, there he is. Okay, he has a polar head. Polar meaning hydrophilic, sorry. And then it has non-polar tails, which are hydrophobic. Okay, and there's two rows. There's this one. This is an entire row of phospholipids all the way across the entire cell. Okay, and that's the outside. This is the extracellular matrix. So this is outside of the cell. Okay, and notice that it has another layer of phospholipids. And the hydrophilic heads are in this direction. So they align with one another because inside the cell is primarily water. Okay, and then indeed the hydrophobic tails align. Now this happens by nature, naturally, because these are lipids. These are specifically phospholipids. Now all of these polar heads are always going to align, and the hydrophobic tails in this region, because they fear water, will always flip towards each other by themselves without any energy expenditure, and this is how all phospholipids are designed. Okay, so let's be very clear here. I want you guys to note this. This region, this is the hydrophilic head. Let me erase that little part. Okay. Hydrophilic head. Okay. And of course there's one on each side. And then this center here. Okay, these are the hydrophobic tail. Really, it's tails, they're plural. All right, either way, the point is this whole outside region, okay, with these blue hydrophilic heads, they align and they can interact with water freely, and they're happy there. And same on this side. But these hydrophobic tails they fear water, they hate water, and they immediately flip and reorganize themselves in this position. Okay, so therefore we have a hydrophobic core. This whole core is hydrophobic. Okay, the center meaning. And the outer portion of the membrane is hydrophilic. And really it's because of the properties of the phospholipid itself. Okay, now there's more. Okay, now this is very important. These proteins in particular, see how this is a protein here. Okay, this little guy. And see how he's embedded all the way through. And notice this protein over here that is not embedded all the way through. Okay, the reason why this particular protein can be embedded all the way through is because it has both properties. It's hydrophilic on each end. Okay. And so down here, this end is also hydrophilic. And it has a hydrophobic, this thing is hard to move around. All right, you guys get the point. Okay, and then it has a hydrophobic core, so the center is hydrophobic. So this allows the protein to be embedded from end to end. Okay, therefore, these are specialized membrane proteins, and they're specifically called integral proteins. Okay, now notice the ones that do not embed through the membrane, they don't have those properties. These are just hydrophilic, so they can hang out over here, no problem. These are called peripheral proteins. And if you look very, very close, you see this little 1, 2, 3, 4 fused ring component. That is cholesterol. And it sits perfectly fine in the hydrophobic region because it is indeed a lipid. And lipids are all hydrophobic. Remember that from the previous chapter. All right, now check this out. See these little sugar-like structures? And they are sugar. But they're not just sugar. They're a hydrocarbon glycoprotein chain. And so in short, you just can learn them as a glycoprotein, which just means a sugar protein. Okay, so if you see these little chains, all of them are glycoproteins. All right, so because of the fact that we have this elaborate display of different properties, like the phospholipid themselves that make up the primary component of the plasma membrane, integral proteins, peripheral proteins, cholesterol, the glycoprotein. That is why we call this the fluid mosaic model. And ultimately, you want to know the full anatomy of all of these components. All right. Now let's look closer at the phospholipid bilayer. And earlier I mentioned that these phospholipids align, and they do. So let's say I grabbed a bunch of phospholipids, all of these, and I tossed them in a bucket of water. They would immediately repel water. The hydrophobic tails would all align and face one another. And the hydrophilic heads could just exist this way and face the water molecules without a problem. okay, and the hydrophobic tails will always align with one another, and therefore the bilayer, the phospholipid bilayer, the plasma membrane, it's made naturally without any energy or help. Okay, now really quick, let's look at this. This is called a phospholipid bilayer, and it's called a phospholipid bilayer because it's made out of phospholipid. And then there's two layers. Here's layer 1 on top and layer 2 on the bottom, and bi means 2. Okay, phospholipid bilayer. All right, what about things that can come into the cell and out of the cell? This is a great diagram for looking at those properties. Now, here's our phospholipid bilayer. Here's some proteins. Okay, really quick, this is a water channel, and I'm going to label it because for some reason the book has not labeled it. Okay, these are called aqua porins, like aqua, like water, right? And then pour, like pour. Very easy. All right. So a lot of the times inside of the books, the diagrams, they won't show you the aquaporins. They just won't draw them because I guess they expect you to know that they're there. So if water can move through a plasma membrane, it is because there are aquaporins specifically there. Now, water does pass through the plasma membrane. Okay, it can go in and out. It follows certain laws, but it can go in and out. Okay, other things that can move in and out. Non-charged molecules. What does that mean? It means they don't have a charge, like this, like plus or minus, no charge, and they have to be pretty small, actually. All right, so what cannot enter or exit the cell with well, let's just put it this way. It cannot enter or exit directly through the plasma membrane. It's going to need an assistant or some help, okay? For example, macromolecules, they are far too big. So you see how this arrow sends it backwards? Same thing over here. It comes near the plasma membrane and it's sent backwards in a boomerang, meaning it can't pass, okay? If something is large, if it's charged, if it has a positive or negative, if it's ionic, if it has a positive or negative charge, it cannot get through the plasma membrane. It will get bounced back. All right, so what can get through? Again, water, small, non-charged molecules can move through. What cannot get through? Ions, charged molecules, macromolecules. Okay, very good. Now, what if something needs to get through the cell and it has a charge? What then? Okay, now that's where these guys come into play. We have proteins that are embedded into the membrane. Remember, we called them integral proteins earlier. Now, to be more specific, we have one called a channel protein, a carrier protein, a cell recognition protein, a receptor protein, an enzyme protein, or enzymatic protein. Okay, a channel protein allows ions to pass through. Okay, now this is very important because it's not just any ion that can pass through any channel. It's very specific. So let's say we're dealing with chloride ion. And all of these little molecules right here are chloride. Let's just use their example. Okay, every single one of those green little guys right there, that's a chloride ion. All right, now this channel is very specifically a chloride, okay, ion channel protein. All right, therefore, every single ion has a ion channel. Let's say that this was something else. Let's say this is sodium ion. Could it move through a chloride ion channel? Does that even make any sense? Absolutely not. No. Indeed, it would have to move through a sodium ion channel to get through. Therefore, every single ion in your body has an ion channel that is designed specifically for it. Calcium ion, and so on. All right, now in this example, they are talking about chloride ion, so let's actually cover that because this is pretty interesting stuff. So this might sound like something that's just far-fetched, way out there in science that doesn't have anything to do with you, but that's not true. Actually, in fact, your chloride ions are absolutely required to operate perfectly, constantly. Because if they don't, let's say there's a defect, then that's a huge disorder, like cystic fibrosis. Okay, for example, cystic fibrosis is a genetic disease. So people are born with it, unfortunately. And what the disease entails is this. It's a faulty chloride ion channel. A faulty chloride ion channel, which means that these chloride ions actually can't really move through the channel very well, or not at all, or they're limited in some way. Now, this is a genetic problem, like I said, but the fact is that cystic fibrosis, it causes one tiny little defect, a defective ion channel, chloride ion, and that person has a terrible disorder. They have a hard time breathing. This mucus collects in their airways, and it in turn affects their entire body. If you can't breathe right, if you have anything blocking your airways, that's going to affect your entire body. And that's why this stuff is so important, because it does matter, and it matters to you greatly. If you don't have cystic fibrosis, that's great. And that also means that all of your chloride ion channels are fully functional. Yay. All right. Next is a carrier protein. So if a molecule is too big, like a macromolecule, like glucose, for example, it's going to have to be carried across the membrane by a carrier protein, just like that. Easy. Cell recognition protein, C. All right, see these little sugar molecules right here? This is called a MHC, a major histocompatibility complex, the MHC complex. All right, now what's interesting about the MHC complex is every single person has a completely unique MHC. That means I have my own, you have your own, and that allows for every cell in your body to know that they belong there. Your cells can communicate with each other, and this is one of the ways. They let each other know, okay, you're okay, you can stay here. That's okay, you belong to this body. Now, why is that relevant? Because we do organ transplants sometimes. Some people need a heart transplant or a kidney transplant. Now, when a person is receiving an organ, let's say they're getting a heart, okay? A heart transplant. Now, that heart is going to have a completely different MHC. They're going to have the donor MHC markers all over those cells. When they go into the receiver's body, what do you think is going to happen? Well, of course, the body is going to immediately recognize that this is not a self cell. It is a foreign MHC, and the white blood cells, your immune system is going to try to attack the heart. It's it will attack it or try to. But of course, we have immunosuppressants and other medications that will stop the body from attacking the organ. And then, of course, that medication has its own issues, and that's a different story. But the point is that every single cell has cell recognition proteins with the MHC complex that is unique to your body. Okay, then we have receptor proteins. Receptors are pretty cool. Basically, what is interesting about them is this. Okay, so they have this very specific shape, and this molecule will bind here. And then it can elicit any response depending on what this molecule is and what the receptor is. So, for example, growth hormone bind receptors. You have neurotransmitters that bind receptors. And let's say a person doesn't make enough of growth hormone. Okay, that's the example they're using. They can actually facilitate that in the doctor office, so we can inject growth hormone into a person so that they can fully develop, or at least develop more so than if they were left to their own proceedings. And basically, the growth hormone that's injected will it will mimic the shape of the natural growth hormone and elicit the same response. Okay, that's what receptor proteins do. A lot of our medications, a lot of them, mimic receptor proteins, receptor molecules. Okay, next is enzymatic proteins. We're going to learn all about enzymes, but some of these membrane proteins are enzymes, which means that they catalyze reactions. So what's happening here is this guy comes in and he's turned into something else. This is a catalytic reaction. Okay, now this is kind of reviewing what we just talked about, the different proteins, the enzymatic protein, the cell recognition protein, there's the MHC complex, the channel protein, carrier protein, and so on. All right, the next thing we're going to talk about is transport, membrane transport specifically. So what we're looking at is the different types of possible methods of moving things in and out of the cell. Okay, the first thing that we're going to look at is diffusion. Now, notice that diffusion is falling under this big bracket called passive transport. Okay, so everything under here is passive, which means it doesn't require any energy at all because it is passive. Now over here we have active transport, which we will discuss later. Okay, the first thing is diffusion. All right, so diffusion is when a solute or a molecule, now this term solute, okay, is non-specific. So this is when a solute moves from a higher concentration to a lower concentration until equilibrium is met. That is what diffusion is. That's what it means. So let's explain exactly what that means. All right, let's look. So here on this side of the membrane, okay, let's just say this is the outside. Outside of the cell, this is the inside of the cell. And we're looking at this red solute. Where is there a higher solute concentration? On the outside or on the inside? Well, all we have to do is look. It's very simple. So if we look inside, there's 1, 2, 3 solutes. If we look outside, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, so by the laws of diffusion, if a molecule can pass through the plasma membrane, it will always move from a region of high concentration to a region of low concentration until they're equal on both sides, which means that these molecules are just going to keep coming through. And I know this isn't perfect, but just imagine that these are solutes until they're equal on both sides. 1, 2, 3, 4, 5, 6, 7. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, now they're equal. Now they're at equilibrium. Okay, now that is how diffusion works. I'm going to show you guys some videos to better explain this and show an animation. But essentially that is the point. Okay, so next. Now, what if something can't actually pass through the membrane? Remember, like an ion, for example. It's going to need something called facilitated diffusion. Okay, so a good way to remember it is that it needs a facility to diffuse, and the facility is this protein. Facilitated diffusion, but it'll follow the same laws. All solutes will move from a region of higher concentration to a region of lower concentration until they meet equilibrium. Okay, so diffusion is when solutes pass through the membrane directly. Okay, facilitated diffusion is when there's a protein involved that's facilitating the transport or diffusion, and solutes always move from a higher concentration to a lower concentration across a plasma membrane until they reach equilibrium. All right, now let's look at the next thing. Osmosis. Okay, osmosis is very specific and very special because it is talking specifically about water. So water moves as well. Water will always move towards the higher solute concentration. Okay, so in this case, these are the water molecules, right? These little blue guys right here, these are water. And these solutes are pretty big. There's 1, 2 solutes on this side of the membrane. How many are on this side? 1, 2, 3, 4. Therefore, water will always move towards the higher solute concentration. Very good. Now, all three of these, diffusion, facilitated diffusion, osmosis, are all passive transport. Now, what if we want to move something against its concentration gradient? Now, in this case, let's look at this picture. The laws of diffusion say that molecules, solutes, will always move from a region of high concentration to low concentration. So naturally, these guys would move which way? Would they move in or out of the cell? Okay, they're supposed to move in. However, there are times where the cell doesn't want them in. It just doesn't. There's different reasons for it, and it's going to have to pump it out. Let's say that it doesn't want these guys pouring back in here following the laws of diffusion at all. Okay, it's going to push these out, move them against their solute concentration from low to high, which is against nature. That's going to cost you. That's going to cost you ATP, which is pretty much money. This is cellular money anyways. All right, so passive transport is when there's diffusion across the membrane. Okay, and it requires no energy. And we can have regular diffusion, which is just straight through, or we can have facilitated diffusion, which requires a transport protein such as this one in this picture, but the fact is it requires no energy. Then we have active transport. That is moving something against its concentration gradient. So this is moving a solute against its concentration gradient. Okay. It requires energy in the form of ATP. All right, so what we're showing here is the solute binding to a specific transport protein. Okay, ATP will have to donate a phosphate out of the 3 that it has. See this phosphate that's here? It will ATP temporarily phosphorylates this pump, this active transport pump, and this pump will move solutes against their concentration gradient. Then the phosphate goes away. It detaches. Pretty simple, actually. Now, osmosis. Osmosis is the diffusion of water. This is specific to water. Okay, and it's the movement of water across a membrane, and it's going to move based on concentration differences on either side. All right, so what we're showing here, okay, now water will always move towards the higher solute concentration. And what they've done is they've stuck a crystal dye block in this fishbowl. Okay, now that crystal dye is made out of these little tiny molecules. Let's look at it. Okay, molecules. This whole bowl is full of water molecules. Water will always move towards a higher solute concentration, so it's going to do that. And that's what's happening here. Water's diffusing towards the crystal dye molecules, okay, until it has reached equilibrium, until the water molecules and the dye molecules are equally distributed throughout the whole entire area. Okay, that's what water does naturally. Okay, we're going to watch a couple of animations on osmosis and osmoregulation.
Have you ever heard of the ick? The disease, that is, that can afflict aquarium fish? I will never forget the first time my fish caught ick. Well, it's called ick. It stands for this. So, ick it is. It's actually caused by a protist, a parasitic one. In our ecology video, we talk about parasites and how parasites benefit while causing harm to their host. And in my case, it was all of my beloved fish. Well, okay, technically it affected Gertrude first, but then it spread because it is a contagious disease in fish. It typically causes these white dots. It gets on their fins, on their bodies, on their gills, all over. It can even be lethal if not treated. So it's bad news, and being a guppy enthusiast, I was really worried about all of my guppies. This is not a video about parasites. This is a video about diffusion. So you're probably wondering, how is she going to connect diffusion to this parasitic protist disease? Well, it's because of the treatment. You see, there is a commonly purchased treatment for the disease of ick. One common treatment option contains an anti-parasitic called methylene blue. Its name is not misleading. It is quite blue. The directions on the back of the medication outlined how many drops to add per gallons of water. And I watched and tried to encourage my fish to swim towards the medicine. But they were panicked, maybe because a portion of their water was changing colors, but also probably because of the kid trying to get their attention to swim towards the medicine. But you know what I didn't know then? I didn't need them to swim over there. The blue drops would spread on their own in a process known as diffusion. Diffusion is when the net movement of a substance travels down its concentration gradient. That means it moves from a high concentration to a low concentration. When the methylene blue drops were added into the water, they didn't just sit there. The molecules traveled in the water through diffusion. The high concentration of molecules, which is where they were dropped, spread to areas of low concentration in the water. Pretty soon, the water had a uniformly blue appearance as the molecules eventually are evenly dispersed. This was very important for treating my fish because the treatment reached all of them, and they soon got better. Diffusion doesn't just happen in water. For example, it occurs if you spray an air freshener in the air. The net movement of molecules travels from an area of high concentration of the air freshener to a low concentration of the air freshener. Pretty soon, it's likely others would smell the air freshener, even if they were quite a distance away. Now, I want to mention two important points. First, we say net movement because that's the overall movement. But it doesn't mean that the molecules can't move around the other direction, and it doesn't mean that the molecules eventually stop moving either. The molecules are continuously moving, even when equilibrium is reached. It's just the overall movement, the net movement, is from a high concentration to a low concentration, until equilibrium is reached. Second, diffusion is a passive transport. That means an input of energy is not needed. We say that passive transport does not require added energy. When we talk about diffusion of molecules in cells, for example, the diffusion of oxygen molecules into a cell, it is classified as passive transport. A concentration gradient is itself a form of potential energy. Passive transport is different from other processes that we detail in our cell transport video, like active transport, where an input of energy is required. And speaking of our cell transport video, we outline that there is also a type of diffusion called facilitated diffusion. Facilitated diffusion is when molecules still have a net movement of a high concentration to a low concentration, but they may be too large or have other characteristics that prevent them from directly traveling across the selective cell membrane. Therefore, they have to go through a protein channel. It's still diffusion because it's still passive transport and it's still traveling down its concentration gradient. It's just that, in that case, it generally has to go through a protein to get inside. Now, there are factors that can affect the rate of diffusion. We'll mention just a few of them when considering these small molecules here, diffusing in simple diffusion. Distance. The greater the distance that needs to be traveled, the slower the diffusion rate. For example, you could observe a difference in a 5-gallon tank versus a 55-gallon one. Temperature. Okay, really quick, would you think a higher temperature or a lower temperature would increase the diffusion rate, assuming everything else is constant for the point of the comparison? Generally, a higher temperature. A higher temperature generally would mean there is more movement of the molecules, so the diffusion rate is increased. This has to do with energy, so check out the link in the description for more about that. Characteristics of the solvent. For example, is the solvent very dense? That could slow the molecules down and decrease the diffusion rate. Characteristics of the molecules, or whatever substance is traveling through diffusion, since technically not everything that travels in diffusion is defined as a molecule. What is the mass of the substance traveling? Generally, a substance with greater mass will have a lower diffusion rate when compared to a substance with less mass. Characteristics of the barrier, if diffusion is crossing a type of barrier. To explain that, let's consider diffusion across a cell, where the barrier would be the cell membrane. Small, nonpolar substances pass through a cell membrane easier than something large or polar. That will affect the rate of diffusion. The surface area and thickness of the cell membrane also will alter the diffusion rate. For example, a large surface area and thin membrane would generally allow a faster diffusion rate than a smaller surface area and thick membrane. By the way, there are more factors that can affect the diffusion rate than just those 5 we listed. In fact, you may have already thought of another factor when seeing our simple diffusion image. Generally, if I were to increase the concentration of the substance here, that would cause a larger difference between these concentrations, and that would also increase the rate of diffusion. Before we end, you might wonder, why should I care about this diffusion thing? Okay, other than the aquarium fish that I love, realize that diffusion is critical for all of life. Cells are the living unit that builds up all life, and materials that cells need to survive have to get in, and waste molecules of the cell have to get out. Now, while diffusion is certainly not the only kind of cell transport, diffusion is responsible for many critical items. One example? As you sit here right now, diffusion is responsible for oxygen leaving the alveoli of your lungs to your blood. Likewise, the waste gas carbon dioxide is able to exit your blood to the alveoli, a critical process made possible by diffusion. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So the next topic is not unrelated. It's called osmoregulation. And now I want you to pay close attention to what we have here. And normally in the classroom, I draw this on the board and everybody draws with me. But of course, we're going online. So it would be wise for you to draw this in your notebook. But that is, of course, up to you. But this is what I recommend. Okay, so let's begin. So what we're looking at here is, well, let me just define this really quick. Osmoregulation. Osmo means water. Think of osmosis, osmo, that means water. Okay, regulation, of course, means regulation. So this is how water regulation occurs in 3 different scenarios. So what we're looking at is, this is a beaker, okay? Beaker, beaker. There's 3 of them. And of course, inside, this is fluid, okay? Beaker solution. And we also have a cell. Okay, this is a very generic sketch, but it's very efficient. Okay, this is the cell membrane, cell membrane, cell membrane. All right, each cell has the following solute concentration: 30% solute, 70% water. 30% solute, 70% water, 36% solute, 70% water. Now, we have placed these cells, okay, in 3 different solutions. One is called a hypotonic solution. One is called an isotonic solution. And one is called a hypertonic solution. Okay, so now, the laws of osmosis say that water will always move across a membrane from a region of the highest solute, the region with the highest solute concentration. So we're going to keep this really simple because there's no reason to complicate this. First simple thing. A solute can be a range of molecules. It can be glucose. It can be salt, NaCl. It can be iodine. It could be different things. Okay, so solute is an ambiguous umbrella term that goes for a variety of molecules. Now, if we look at the hypotonic solution, our cell has a 30% solute concentration. The rest is water. Now, the solution inside of the beaker has a 20% solute concentration. Now, according to the laws of osmosis, water will always move towards the higher solute concentration across a membrane. What's higher, 20% solute or 30? Okay, obviously it's 30. Therefore, which way will water move? Will it move into the cell or out of the cell? Okay, if you said into the cell, you are correct. Therefore, water is going to move into the cell. If you place a cell in a hypotonic solution, what does hypo mean? Okay, hypo means less. The solution we're discussing is this one, the beaker solution. Okay, it's got less solute. That's why it's called hypotonic. Okay, water will enter the cell. However, this is not necessarily a good thing because water flowing into the cell can cause something called cell lysis. Remember lysis? Lysis means to break. So this cell can be broken. Okay, next, isotonic solution. Now, Iso means same. And if you look at the solute concentration, the cell has 30% solute, and so does the solution. Therefore, there is no change in the water movement. What about a hypertonic solution? Okay, remember the word hyper? Okay, that term, what if someone's really hyper? Okay, they're too much, right? Or they've had too much sugar. Or someone that has hyperthyroidism. Their thyroid is overactive. Okay, bottom line, hyper means more. Now, indeed, the beaker solution has a 40% solute concentration, and 40 is indeed higher than 30. So which way will water move? Water always moves towards the higher solute concentration. So will water enter or exit the cell? Water will exit the cell. Water will leave the cell, and the cell will actually shrivel up and shrink. Okay, these are the 3 scenarios of osmoregulation, and yes, you have to know them, and yes, you will be tested on this. All right, draw, memorize, learn, all of it.
Surrounding a cell is isotonic with the cell. That means the solute concentration is the same outside the cell as it is inside the cell. While water is diffusing into the cell from the solution, and while water is diffusing out of the cell from the cytosol, the rate at which these two processes occur is about the same. And so therefore there will be no net change in the volume of the cell, since the amount of water entering is about the same as the amount of water leaving. This is what occurs when the solution surrounding a cell is isotonic. When a cell is in a hypotonic solution, the solute concentration around the cell is less than what it is inside the cell. In fact, this hypotonic solution may even be pure distilled water, which would have no solutes whatsoever. Since water goes to where the solute is most concentrated, and since while there are solutes in the cell, there are fewer, if any, solutes outside the cell, this means that the water from the environment goes into the cell. The cell will tend to swell as it takes on this additional water. Especially if a cell is placed into distilled water, which has no solutes whatsoever, the rate at which the water enters the cell is so great that the cell swells to the point where it explodes. All of the contents of the cytosol spill out, and then an empty cell membrane is left behind. And so when a cell is in a hypotonic solution, water enters the cell and the cell may swell or even burst. When the solution around a cell is hypertonic compared to the cell, hyper means too much, and a hypertonic solution has a higher concentration of solute than one finds inside the cell. Since water will have a net rate of diffusion towards where the solute is most concentrated, and since the solute is most concentrated out of the cell, this means that water will tend to leave the cell. This can actually be observed because as water leaves the cell will shrivel, a process known as crenation. And so therefore, in a hypertonic solution, cells will shrivel or undergo crenation as water leaves the cell.
Okay, so this is sort of a little recap of osmoregulation. The only difference here is we're comparing animal cells to plant cells. Okay, animal cells, these are human red blood cells. Okay, they prefer to be in an isotonic solution. Okay, and you can see water coming in and out, but it's coming in and out at an equal rate. Plant cells do not like that. In an isotonic solution, a plant will be flaccid, limp, no good. All right, now let's look at a hypotonic solution. So in a hypotonic solution, water rushes into the cell With an animal cell, it's going to cause cell lysis, which means it'll rupture. Okay, with a plant, that's not going to happen. This is indeed the ideal environment for plant cells because they have the central vacuole that can store water, plus they have a cell wall, and it can't rupture. It will, however, inflict pressure on the cell wall, and that pressure is called turgor pressure. Okay, now in a hypertonic solution, water leaves the cell, and with an animal cell, the cell will shrivel up. That's not good either, but it happens. Now in a plant cell, we have something very specific that occurs. It's called plasmolysis. Okay, the plasma membrane lyses or breaks away. See how it's pulling away from the cell wall? And actually everything is going to move to the center. And that is plasmolysis. That's what happens to a plant cell in a hypertonic solution. Okay, now, what if a molecule is too large to cross the membrane in any way? What if it can't get through an active transport protein, a carrier protein, channel, none of that? What if it's big? What if it's huge? Well, we have solutions for that. Okay, then the cell will use a methodology called bulk transport. All right, these are important terms. Endo means inside, and cytosis is in reference to the cell. Now, these are important things to memorize now, though. Okay, endo equals inside, okay, or innermost, like the innermost region of something. Okay, now exo, on the other hand, think of the word exit. When you see an exit sign, what does that mean? Obviously, that means you can leave out. Okay, so that's either outside or outermost. All right. So endocytosis is inside the cell, and it's a method of transport that allows cells to take in huge or large substances by creating a vesicle. And I will show you what that looks like in upcoming slides. What we are looking at here is the other one, exocytosis. Exocytosis allows the cell to move large substances like proteins, waste. It can move out antibodies, which are technically proteins. It can move quite a bit out of the cell. It can move fluids out. And the way that it does so is as follows. This is called exocytosis, and you've seen it before, actually. Whether you remember or not, you saw it in chapter 4 at the very end of protein production. When a cell makes a protein, it goes from the Golgi apparatus to the plasma membrane. This is how it does it, through exocytosis. So our cells can basically compartmentalize molecules, which is what's happening here. And notice, if you look closely, it's made out of phospholipids. It's made out of a phospholipid bilayer, layer 1, layer 2, molecules on the inside. Okay, all it has to do is interact and bind with the plasma membrane, and slowly but surely the phospholipid bilayer in this vesicle will become part of the actual plasma membrane itself. And it will continue to do so till all of these phospholipids integrate with the bilipid layer, thus releasing all of these molecules outside. That is called exocytosis, and it's a method of bulk transport, and it's pretty common. Now, endo means inside. There's 3 types: pinocytosis, phagocytosis, receptor-mediated endocytosis. Pinocytosis, okay, this is referred to or nicknamed cellular drinking. Cellular drinking. Why? Because the cell is kind of drinking. So here's the inside of the cell. Here's the outside. When a cell wants to bring in fluid, it will make this little pit. It makes a little dimple. And the two sides of the dimple are going to get closer to one another until they have completely cleaved off into its own vesicle, bringing with it all of this fluid. Thus, we get the word penocytosis, which means cellular drinking because it's bringing fluid in. Next, phagocytosis. Phagocytosis is when it's referred to as cellular eating because solid molecules can come in. All right, now all cells can do this. And basically when the cell encounters something that it wants to take in, like food, it will make that little pit, and the pit will continue to deepen, and the two ends will get closer until they've completely clipped off a vacuole of food, and that is phagocytosis. Receptor-mediated endocytosis is basically this, pretty straightforward. Receptor-mediated endocytosis is when there's a receptor and a specific molecule involved. Okay, and basically the endocytosis will not occur until the molecule has bound to its receptor, and then it will initiate this whole process of endocytosis, bringing into the cell a vesicle with the receptor and molecule and anything that was in the way. And that's that. Pretty straightforward. This is all in reference to bulk transport. And think bulk means bulky, and that's the bottom line. Now I am going to show you guys some an example of phagocytosis that's pretty interesting. It's a white blood cell, which we have in our body, and we get to see a white blood cell under the microscope destroy a bacteria through phagocytosis. All cells do this. Your cells do this, and so do amoebas, and so do white blood cells, which are technically human cells and animal cells in general. All right, so let's check that out.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Okay, the next part of this chapter is metabolism, energy, and enzymes. So this is the second section of chapter 5, but it's still chapter 5. Now, let's start from the very beginning. So we need to talk about energy and basically how energy works, and then we will go from there. So basically, energy is this. It's the ability to do work or bring about some kind of change, like what we saw earlier with active transport. Okay, so for example, if you need to lift something, that is going to require energy, the ability to do work or bring about some change. And you are doing work. You are lifting something, and that requires energy. Okay, now our cells use energy in the form of ATP. And we're going to talk quite a bit about ATP as we go further into this class. All right, so we need a constant supply of energy, and basically everything on this planet needs a constant supply of energy, everything that's alive. Okay, now how do we get that constant supply of energy? Now, you might remember this from chapter 1, but all of our energy comes from the sun. Okay, and basically we end up getting it through our diet, and other organisms do as well, and we need it for growth, metabolism, and reproduction, cellular reproduction, and other reproduction. Okay, so whenever we talk about energy, we have to talk about thermodynamics, because the laws of thermodynamics explain energy and how it works in a couple of different scenarios or laws, basically. But ultimately, it begins with the sun. All energy, all of it, comes from the sun. Okay? And it is picked up by photosynthetic organisms, like a tree, for example. Photosynthetic organisms can convert solar energy from the sun into this form, chemical energy. Okay, an animal will eat through their diet, they will acquire that chemical energy. If you eat vegetables, if you eat meat, it doesn't matter. As long as it's either a plant or animal or something that has some kind of physical anatomy carrying chemical energy, that is going to work. All right, either way. So solar energy is from the sun. It's converted to chemical energy by photosynthesis. And then animals acquire that energy in their diet, and that chemical energy is transferred into mechanical energy. Mechanical energy is energy that's being used right at that moment. Energy in motion. Okay, so this moose is walking, right? And he's able to do so because of the fact that he has quite a bit of chemical energy in his body from eating, and his cells are able to utilize this chemical energy to bring about mechanical energy. Okay, so all of the life on Earth is dependent on solar energy. So let's say this is kind of an unrealistic scenario, but I don't know if you guys watch The Simpsons or not, but I don't know if you've ever seen, if you do, there's an episode where Mr. Burns wants to block out the sun because, of course, you know, he's this ultimate villain or whatever. Now, it's interesting because if the sun were to be blocked out, everything on the planet would die. Everything. It might take a while, but everything on this planet would die. And that's actually what wiped out the dinosaurs. An asteroid hit the planet, okay, and it kicked up so much debris around the planet and blocked out the sun. Okay, you block out the sun, and okay, no sun. The first thing that's going to die are the plants. There's no plants to make chemical energy, and there's no plants to make oxygen, and then it's just a domino effect after that. And that is how the dinosaurs got killed off. It might have taken months for them to die completely, but or for all of them to die. It might have even taken close to a year or more. We don't know exactly, but this event did occur. Okay, so either way, that is the significance of it. Let's go ahead and move on. Now, when I said chemical energy, okay, that's found in plants, that chemical energy has another term, which is potential energy. Okay, and we also talked about mechanical energy, and what it really is kinetic energy. So I want you to understand that these two are basically the same thing, energy and motion. So the two types of energy, according to the laws of thermodynamics, is potential and then kinetic. Potential, think of potential. What is something that has potential? It has the ability to do something, right? In other words, it's stored. It's stored energy. Okay, kinetic energy, kinesiology is the study of movement and motion. So kinetic energy is energy in motion. So here in this picture, you see this cheetah. Cheetahs are fast. And as this little guy is sitting here, he's not doing anything yet, but he has the potential to run incredibly fast. Okay, so when he's in this position, the energy that's stored in all of these muscles, that's his potential energy. And all of the energy that he has in his sensory organs and so on, right? And then when he takes off and he's running, that is energy in motion, kinetic energy. That's as far as it needs to go in this class. All right, now, there's three laws of thermodynamics, but you guys are going to learn two laws of thermodynamics. The first law is called the law of conservation energy, which says energy cannot be created or destroyed. And that's true. It can't be created or destroyed. However, it can be changed from one form to another. That is true also. Let's see. Now, side note, that's the primary part of the law, but side note, energy, every time it's converted from one form to another, we lose some. We lose some energy in the form of heat. So if we go back to this, I know I drew all over it and it's a little bit of a mess, but let me kind of go backwards here. Okay, so if you look at this picture, in your textbook or in the slides. Okay, now I want you to pay close attention to the first law of thermodynamics. It says energy cannot be created or destroyed, right? But it can be changed from one form to another. Okay, that is a law. And the fact is that we can't make energy. The energy we have, the energy sources we have on this planet, we didn't make them, right? We didn't make coal. They mine coal. It was already here. We don't make gasoline or petroleum oil, basically, which is turned into gasoline. It doesn't matter. We don't make it. It was already here. And if you were lucky enough to be born into some oil tycoon family, good for you. But for the rest of the normal people that are not oil tycoons, we don't have the luxury of owning something so rare with that kind of life source and that energy source, because that's not how energy works. Energy cannot be created or destroyed. It comes from the sun, and it's transferred into chemical energy. And it can be transferred into mechanical energy, and it can be transferred into cellular energy. Okay, now this is putting coal and all that stuff to the side. That stuff, these are organic compounds that took, millions, hundreds of millions of years to develop. So that's a separate thing, and I'll discuss it in later chapters. But the most important part is to understand that all energy comes from the sun, all of it. And without it, the earth could not exist, and the earth could exist, we wouldn't exist, and neither would all of the living organisms. All right, I think you got the point. Let's go on. All right, so, oh, no, the other point. My bad. Okay, so every time energy is transferred, we lose some as heat. Every time. Sometimes they'll show it, sometimes they don't. Here they are showing it. Okay, that was a side note. Okay, so let's repeat. The first law of thermodynamics. is the law of conservation energy, which says energy cannot be created or destroyed. However, it can be changed from one form to another. But every time energy is transferred from one form to another, we lose a little bit of energy as heat. Okay, great. Law number 2. This law says that when energy conversions occur, it increases energetic disorder. Okay, so energy disorder or randomness. In other words, this term, entropy. Entropy means randomness, disorder, chaos. Okay. Randomness, disorder, chaos. And in addition, in life, nature, natural phenomenon tend to increase in entropy. Okay, so for example, let's say you clean your bedroom, your room, and everything's spotless. Okay, then over the course of a week, then 2 weeks goes by. Is your room going to be spotless anymore? No, because of the simple fact that the second law of thermodynamics says that in nature, things tend to move from order to disorder, to increased entropy, to increased chaos. That is the way of nature. All right, now let's emphasize our energy sources. So the first one. Now, all energy comes from the sun, and we rely completely on photosynthesizing organisms such as plants and algae and other photosynthetic organisms to do this for us. They make for us something incredible. They make for us food, all of the food we need, and they make for us oxygen. We could not survive without our plants, and they're cutting down our rainforests, but that's a different issue. All right, now the point is that this is a plant cell. Okay, so these plant cells, these photosynthetic organisms, they do not create energy. They take solar energy, plus they take CO2 and water, and they make sugar. They make carbohydrates. Okay, more specifically, yeah, that says carbohydrate synthesis, and that's great, but the book needs to be a little more simple. And more specifically, what this thing is doing is making glucose, and I want you to remember that. Glucose. And glucose is what? Glucose is a form of chemical energy. So that's the energy transformation right there. Solar energy, okay, through photosynthesis is converted to glucose, which is chemical energy. Okay, great. Next. Now, of course, some is lost as heat, yada yada. None is destroyed. Okay, now the other biochemical energy process is called cellular respiration. Now, with cellular respiration, typically cellular respiration is it's used for everything, basically, to make your cellular energy, and it's utilized for everything that is not a spontaneous reaction. All right, so what we're looking at here, this is a muscle cell, okay, and they're showing a carbohydrate going in, okay, but that's also not the full story. So I'm going to cover this with more specific notes, what's actually going to come in here is glucose. Okay, and oxygen. All right, now that is brought into, it's actually already in our cells, but that is used in our cells. And our cells take that glucose, and what they make during cellular respiration is ATP. Okay, it's not easy taking notes on this thing, so you have to bear with me. ATP. All right. And that ATP, this reaction you'll see again and again and again. Okay, that ATP, and actually let me just be very clear, they also make CO2 and H2O. All right, now this ATP is cellular energy, okay? And that's the other energy transfer. So energy is not created or destroyed. It goes from solar to chemical to cellular. That's not the pathway every time, but that's the pathway for us. All right, so ultimately, energy comes from carbohydrates such as glucose, and that glucose is used to make ATP. And ATP is something that will allow your muscles to contract. In fact, you cannot live without ATP. Okay, this is kind of an interesting side note. So when a person, a human or an animal, it doesn't matter, let's just say human, when a human passes away, when they are dead, at some point after their death, the body goes through something called rigor mortis, and that's when the body becomes completely stiff. Okay, why does it become completely stiff? The reason why is because ATP, okay, is used to contract and uncontract muscles. And when a person is dead, there's no more ATP to uncontract the muscles. So all of the muscles contract and they become stiff because there's no more ATP to uncontract the muscle. Okay, just fun side note. But the point is here is energy has been transferred multiple times, not created, not destroyed. All right, very good. Let's move on. The second law. All right, the second law is about entropy. Now remember, entropy is chaos, randomness, disorder. And the second law states that all matter, remember matter from chapter 2, okay, moves towards disorder, and that's true. So what we're looking at here is glucose, and it's a ball and stick model, and I want you to actually learn this formula and know it for the rest of the semester. This is the formula of glucose, and let me actually, that's a terrible box. Okay, the formula for glucose is 6 carbon, 12 hydrogen, 6 oxygen, C6H12O6, glucose. All right, this is the ball and stick model of it. Glucose is a very organized, high energy molecule. It has tons of potential energy. Okay, it has tons and tons of it. But it has low entropy because it's not disorder and chaos. It's highly organized, right? So it's the opposite. So it has low entropy. And in fact, because this is kind of the opposite of what nature wants to do, it's unstable, and it's less stable than these guys over here. So basically, we know that we get energy out of this. When glucose is broken down, energy is released. And that was the energy that we just talked about. ATP is released. Okay? And the byproducts are water and CO2. Water and CO2 are super small molecules. They're not like this. They're not this big, fancy, organized compound like glucose. They're really not, they're important, but they don't have a lot of energy. They're disorganized, and they're more stable, and they have high entropy. Entropy is this chaos right here. This mess, I guess you could call it. Either way, that is the second law of thermodynamics. Things in nature tend to move towards disorder. And in this particular situation, glucose just happens to be a high energy molecule. All right, so let's look further at glucose and cellular respiration. Glucose and oxygen are absolutely required for aerobic cellular respiration. Okay, glucose is an organic compound, and it is a high energy molecule. And remember, glucose is a C6H12O6 molecule. Oxygen, of course, is just O2. Okay. Now, we get glucose and oxygen from, well, we get glucose from our diet, and we get oxygen from our respiratory system. We breathe this oxygen in. This all goes to your cells. Every single cell in your body receives oxygen around the clock, all day long. Why? Because you have to have it. You have to have it in every single cell. If a cell is cut off from oxygen, even for nanoseconds, it can die. All right, so now glucose is brought into a cell, and it's going to be broken down through cellular respiration and to produce ATP. It's transferred from that cellular or that chemical energy to cellular energy, and ATP is used to do cellular work. Heat is given off, and the byproducts are carbon dioxide and water. Okay, so ultimately that's how every single cell on the planet that's aerobic, that uses oxygen, that lives off of oxygen, that's how they operate. That's how you operate. That's how every single one of your 30 trillion cells operates. All right. Now, what's interesting is that a car runs very similar to the human body, to cells, to living organisms, and it's very much not alive, but it does. Instead of glucose is gasoline, and gasoline is this organic compound, and oxygen is required as well. And we put those together. We put our gas in, start your engine. There's some kind of, I don't know a lot about cars, but I think it's called a catalytic converter or something like that. I don't really know. But the point is there's a combustion reaction, and we get kinetic energy, and your car drives. Okay? And it gives off heat. That's why cars get hot after being operated. And what else? Carbon dioxide comes out of the exhaust, and water. And what are our byproducts for cellular respiration? Same, carbon dioxide and water. Pretty interesting, okay? But what's, I guess, keep that in mind, but also this is potential energy, the gasoline. And so is this. Okay. Now, the next part of this is looking at biochemical reactions, and a lot of the time you'll see biochemical reactions written out, and This is a non-specific reaction, but you need to know it and write it down, even though it's in the notes. You guys should be writing down as much as you can. Now, it says A plus B, there's an arrow, makes C plus D. Everything on this side of the arrow are your reactants. And just leave it at that. You don't have to make it complicated. It doesn't need to be more complicated than that. Literally, everything to the left of the arrow is going to be known as your reactants. And yes, you will identify reactants often. Okay, everything on this side of the arrow are your products. So we're going to look at some biochemical reactions. All right, before we get into the primary ones, I want to discuss two major umbrella. Oh, last time I tried to draw an umbrella, it was terrible. So I'll just do this. Two very big, broad terms. Exergonic reactions, there's my umbrella, okay? And endergonic reactions. All right, so anytime we have a reaction, there is energy involved. Some reactions, exergonic reactions, they release energy. Remember what exo means from earlier? What does exo mean? Exo means exit, means out. All right, so an exergonic reaction releases energy. Okay, so and the reason why is based on this graph. Look closely at this graph, okay? The y-axis says potential energy of molecules, okay? Now we have reactants here, we have our arrow, we have products here, and it's showing the amount of energy. Okay, so what they're telling us is reactants in exergonic reactions are high potential energy molecules. Okay, so when the reaction occurs, when these guys get broken down, okay, we get a release of energy. That's why they're called exergonic reactions. They release energy out. Okay, and then of course we get our products. Products have a lower amount of potential energy, but that's not the point. Okay, endergonic reactions are the exact opposite. The reactants have hardly any energy, hardly any potential energy. And we're trying to build this product that has all this energy, right? Now, that doesn't happen magically. That's not how life is. That's not how energy works. Remember, energy cannot be created or destroyed, and you want to build this high energy product, well, you're going to need to put in some energy input. All right, endergonic. Remember, endo means inside. So just that's why I want you to learn those prefixes and suffixes. They're going to make your life a lot easier in this class. Endo, remember, means in or innermost or inside. All right, so this is, in this case, this means input. Okay, so endergonic reactions, again, there's not technically an O here, but it doesn't matter. They're called endergonic reactions. So anyone that was thinking that, that's the answer. All right, either way. Endergonic reactions requires an input of energy. Okay, so let's recap. Exergonic reactions are a blanket category term for a series, like an endless list of reactions, right? Okay, and these type of reactions, when they occur, they release energy out of the reaction. That's why they're called exergonic. Endergonic reactions are also an umbrella blanket term for this multitude of reactions, but either way, it doesn't matter which reaction it is, it's an endergonic. That means it's going to require an input of energy, big input. Okay, so next we're going to talk about ATP, which is cellular energy. It's also referred to as cellular currency. Now, what's a currency? What's the currency in the United States? Okay, of course, the currency here is the dollar. In the UK, it's the pound. In Jordan, it's called a dinar. In Mexico, it's called pesos. Um, Either way, that is currency. So when you travel, if you go to another country, a lot of the times, one of the things you want to do is switch your US dollars for their currency. And, so you can shop and do whatever you want to do. Not everywhere takes Visa. Actually, quite a few places don't take it, in other countries. All right, so that's currency in our world. Now, in the cellular world, what is money? What is currency? ATP is the most valuable thing in our cells. So they refer to it as currency. Now, what do we use currency for? To get things that we need, right? To buy things. Okay. And therefore, if a cell needs to do something, they're going to have to pay up. And how do they pay up? They pay up with ATP. Okay. Cellular currency. Okay, so what is ATP? ATP is adenosine triphosphate, and this is its, let's zoom in on it. Okay, if you look closely, you see this 5-carbon sugar called ribose right here, and you'll see a base called adenine, and then you see 1, 2, 3 phosphates. Okay, so notice there is a similarity between the RNA nucleotide. For example, ribose is found in RNA, so is adenine. But what's the difference? Is in fact that there's only 1 phosphate here. But either way, they're similar. So this is adenosine triphosphate. Tri, T-R-I, and you probably already know this, but this is a very good thing to know. Okay, tri means 3. Okay, di, D-I, means 2. Okay, sometimes they'll put bi, and that means 2 as well. All right. Now, when we use ATP for energy, for work, to do something, like what we saw earlier in active transport, we need to break off the 3rd phosphate. And there's only one way to disassemble something like this, and that's with hydrolysis. And there it is. 1 water molecule will lyse the 3rd phosphate, releasing the energy. And notice this, when it does so, the phosphate is not destroyed. The energy is released and utilized, but this is not completely destroyed. In fact, it makes this temporary molecule called adenosine diphosphate. Di, 1, 2, plus inorganic phosphate. Okay, and so it's referred to as ADP, and this is cyclical. All right, so that's ATP. Now, when ATP is converted to ADP and that energy is released, what's actually happening is the molecule is being phosphorylated. When something is phosphorylated, like this, or this, or this, okay, that is energy. That molecule is energized. The phosphate will disassemble once it's done what it needs to do, like build a product, or pump solutes, or contract a muscle. And then what do we have left? ADP plus inorganic phosphate. All right, so ATP is cyclical. ADP and ATP is cyclical. And what's interesting is, that how amazing just everything comes together. Now, this happens, this cycle is perfect. It's completely perfect because all of the exergonic reactions that release energy outside exit, right? They in fact provide the amount of energy required to connect this loose phosphate back to the ADP, making ATP. And therefore, all of the exergonic reactions drive ATP synthesis. Okay, and then ATP is used for all of the endergonic reactions. And then it goes on. The ADP is recycled over and over and over and over. This is called energy coupling. Sorry, I crossed that out, but make sure you know that. All right, guys, the next part of this is enzymes. Enzymes are quite fascinating. We've been talking about chemical reactions, right? And now, actually, before I move into enzymes, I'm going to actually do one last thing. And I'm going to do it right here on this screen with you guys. Now, I want you to write this down, notes, okay? I want you to identify, I'm going to put two reactions up here. And I want you to tell me which one is the exergonic and which one is the endergonic reaction. This is a great thing to do, and I normally do this inside of the classroom with the class, but it's a great thing to do in general. So just go ahead and do it at your own house. This should be on your notes. Okay, this is cellular. I'm teaching you some new information. This is cellular respiration. Okay, cellular respiration is actually, let's be more specific. This is aerobic cellular respiration. Aerobic cellular respiration, the chemical reaction is as follows. Okay, it's C6H12O6. That is glucose plus 6 oxygen, O2. And I don't really know how to do I do know how to do it. Okay, so I can, I believe, make this small. Yeah, okay, that's going to take too long. I guess it's fine. Sorry guys, just hang in there. Probably won't do it for the next one. I just want to be very clear. Okay, then you're going to have 6 oxygen. All right, so here we go. Okay, now we need to make this big again, of course. All right, so it's C6H12O6. Okay, perfect. Those are the reactants, and that is something you're going to have to identify, and you do have to know this equation. You should memorize this equation for the rest of this class, basically. We're going to work with it again and again and again. All right, so we have 1 glucose plus 6 oxygen, and what do we get? We get the following. We get approximately 36 ATP. Okay, now this thing is just turned against me. Do what I say. There we go. Okay, we get 36 ATP plus 6 CO2 plus 6H2O. Now, do me a favor at home and go ahead and make these into a subscript and write it correctly. Unfortunately, it's just a little difficult with the typing to get it perfect. It just takes up too much time. All right, now that is the formula for aerobic cellular respiration. You should know that. Now, the next one that I'm going to teach you is photosynthesis. These two reactions are what make the world go round. Okay, photosynthesis is as follows. This is happening in plants, remember, and specifically inside of the chloroplast. And with aerobic cellular respiration, this is of course happening in the mitochondria. So you should remember that from chapter 4. Photosynthesis, of course, is going to be inside of the chloroplast. Okay, here we go. Photosynthesis is, we need 6 CO2. From the atmosphere. Plants can take up CO2. It's a gas. And we need 6 H2O. Plants can get H2O from their roots, of course, right? So no big deal. They get CO2 from the atmosphere. They can absorb it. They breathe in a way, like the way we breathe. We breathe in oxygen. These guys take in CO2. Okay, plus they need something else. It's not over. They need light/solar energy. You can call it either one, but sometimes the book will flip-flop the terms and say light. Sometimes they'll say solar. Really, they're equivalent in accuracy. Okay, and what gets made? All right, what gets made is every round of photosynthesis will make 1 glucose, which is a C6H12O6, and it gives off 6 oxygen, and that is the oxygen that you breathe. And notice this, the reactants of cellular respiration are in fact the products of, okay, why isn't it letting me do that? Okay, either way, are the products of photosynthesis and vice versa. And notice that the products of cellular respiration are the reactants. Now, the question was, which one of these is the endergonic reaction and which one of these is the exergonic reaction? Now, I want you to work that out yourself, and that is something that will put you on a pretty solid path to making these big connections. All right, there you go. Next topic is enzymes. Now, a couple of facts about enzymes. Number 1, enzymes are proteins, all of them. But not all proteins are enzymes. Enzymes catalyze chemical and biochemical reactions by decreasing the activation energy. Okay, the activation energy is the energy required to allow a certain reaction to occur. Okay, enzymes, after catalyzing a reaction, they do not change the end result of the reaction, nor are they destroyed or consumed in the reaction. They're just recycled. Enzymes are pretty interesting. But to emphasize what's happening here, every reaction, see, we're going from reactants to products. This is called the activation energy, okay, and it acts like a barrier. So it's this particular amount of energy that's required for the reaction to occur. So what an enzyme does is when an enzyme is present, it reduces See how it's been reduced drastically from here to here in the presence of an enzyme, okay? It reduces the activation energy, and that is why the reaction happens faster. That's what a catalyst is. Okay, so that's that. Now, if you see a graph, what we're looking at is a graph of two reactions. Here's the reactants, here's the products. And if you look at the left y-axis, it's measuring the amount of energy. And the x is the progression of the reaction. Okay, now the black line represents one type of reaction, and the red line represents a different type. Now, which one is representing a reaction in the presence of an enzyme, and which one is representing a reaction in the absence of an enzyme? You tell me. Is it the black or the red? All right, now the black is very clearly, there's no enzyme. The red, see how the activation energy has dropped all the way to here? This is in the presence of an enzyme. Okay, very good. Now, enzymes are very specific. Each enzyme is only able to bind one type of substrate. So what does that mean? Okay, well, an enzyme is a protein, right? And all enzymes have something called an active site. That's it. Every enzyme has its own active site. This is an active site. And only one substrate, which is in other words a reactant, can fit or bind into here. And they're pretty shape specific and substrate specific. So let's take a look. Okay, so enzymes are specific. What do we mean? This. They can only bind to one substrate and one substrate only. Every enzyme has one substrate. So if this guy tries to bind here, not going to happen. But this one, absolutely, that belongs there. That is the correct substrate. So in other words, it's one enzyme per one substrate per one reaction. And this one, of course, will fit perfectly right there, and that's that. Enzymes are very, very specific. Now, this is an enzymatic example. This is an enzyme called sucrase. Sucrase. Sucrase is an enzyme that breaks down sucrose, and this is its active site. Right there. Okay. Enzyme sucrase. Now, a quick little side note, you're going to see enzymes are referred to as something that ends in ase. A-S-E. All right. So the substrate and or reaction in this situation is sucrose. Remember, sucrose? Sucrose is a disaccharide. Now, when sucrose meets its enzyme, it's going to fit into this particular space. And indeed, it's going to, because it belongs there, the sucrase is going to wrap around it kind of. Okay, I don't know why this is not being cooperative. Okay, so it's going to wrap around it. See how it's changed? The shape from being straight up and down to kind of curved. That is called the induced fit model. Okay? However, we still need to break down a disaccharide, and there's only one way to do that. There's only one way, and that is by using Hydrolysis. There it is. Hydrolysis comes right in. All right. And then we have a temporary enzyme substrate complex. And then our products are released, fructose and glucose. And the enzyme is returned to its natural form, empty, and ready to go again to break down the next sucrose if it finds one. All right, so this is an important formula that you should know and remember. With enzyme plus substrate, okay, they form a temporary enzyme substrate complex. Enzyme is unchanged, and we get products. Each enzyme requires will only work with one reaction. Every reaction has one specific enzyme, and every enzyme has one specific substrate. And usually the enzyme is named after their substrate. For example, these guys right here. The substrate is lipid. Okay, this particular lipid, or lipids are, certain lipids are broken down by lipase. These are digestive enzymes, actually, this one in particular. Urea. Urea is a waste product, and it's broken down by urease. Maltose. Remember maltose? Maltose is a disaccharide. It's broken down by maltase. Ribonucleic acid, RNA. It's broken down by ribonuclease. Lactose. Lactose is the sugar in milk. It's broken down by lactase. Remember the A's thing that I mentioned? Now, that's on all ways. That's just most of them. You will see enzymes that have a different name. All right. Now, there are certain things that affect an enzyme's ability to function, whether it's going to be fast or not. These are the things that affect it. The enzyme substrate concentration. In other words, how much enzymes in the test tube, how much substrates in the test tube. Temperature. Anytime you increase temperature, you're going to increase enzyme activity. pH. pH is very important. Okay, and these are some of the main factors. And then there's some enzymes have to be activated, some enzymes are inhibited, and some enzymes require something called a cofactor or coenzyme. So let's take a look. This is the general effects on temperature. As you increase temperature in regards to a chem reaction with an enzyme present, you will increase the enzyme reaction. So if you look at this graph, anytime you see a graph, you want to analyze it immediately. How do you do that? You look at the X and Y axes and look at the title. The title says effects of temperature on enzymes. The title is the title of the graph. The Y-axis says slow to fast in rate of reaction, and this is temperature. From 0 to 60 degrees Celsius, or 32 to 40 degrees Fahrenheit. Okay, so notice that as we increase temperature, we're increasing the enzyme's reaction rate. Okay, so our bodies, and in general, the ideal temperature for enzymes is 37 degrees Celsius or 98.6 degrees Fahrenheit. So most human enzymes, they prefer this temperature. However, notice this. As it gets hotter and hotter and hotter, what's happening? The reaction's slowing down and actually stopping. Because if it's too hot, what can happen? If it's too hot, the protein can denature. What does that mean? The protein unravels. If the protein is unraveled, the enzyme, then it can't do anything at all. All right, so the general rule of thumb with temperature is as you increase temperature, you're going to increase the activity of the enzyme. However, if it's too hot, you can denature the protein and ruin it. All right, let's go next to this particular animation. So this is representing enzyme activity. So heat increases enzyme activity. Why though? Well, it's because these are molecules and these are microscopic. I'm sorry, they're not even microscopic. They are literally invisible and we can't see them, but we know they're there because we have molecular biology. So we know that they're there, and we have chemistry and organic chemistry. And so usually when we're working with enzymes in the lab, they're a liquid. But what's happening is the enzymes are present and so are the substrates. And the fact is this. These are not magnetically drawn to each other. No, they are bouncing around all over the place. So molecules don't sit still. They shake and they bounce and they move all over the place, okay? They're constantly moving. So these guys are constantly moving. If you increase the temperature, well, you're going to increase the probability that these bouncing molecules are going to collide with one another. So you increase temperature, you're going to increase the rate of collision, and that's just a fact. Increasing the rate of a reaction. All right, the effects of pH on enzyme activity. Now this one's pretty interesting because it's not as straightforward. This is how it works. Each enzyme, has an optimal pH. So an extreme pH can denature an enzyme. However, it's what's extreme for one enzyme may not be extreme for another enzyme. For example, what we're looking at here is a pH scale and the rate of how fast this reaction is going. This is the pH from 0 to 12. And check this out. Okay, this is an enzyme called pepsin. And it peaks at a pH of 2. It has the highest rate of reaction. And what's interesting about that is pepsin is in your stomach. Your stomach has a pH of 2. Your stomach is extremely acidic. And therefore, pepsin is only active, in this pH scale. If you increase pH higher than 3, it's going to crash. And pepsin cannot operate because it's denatured. However, this guy, trypsin, he is also a digestive enzyme and operates best at pH 7, pH 8, because your small intestine is around that pH. And therefore, these are two perfect examples of how each enzyme has an optimal pH. And typically the ones in our body do sit here around this. They're usually around 7, 8, because remember our blood pH is 7.45, no higher than that, 7.35 to 7.45. All right. Other things that can affect an enzyme: cofactors. Cofactors, coenzymes. Now, these are really just not a huge thing that I want to focus on. Basically, it's this. Some enzymes need a helper molecule. They need it. They have to have it. They can't function without it. Okay? And if they do, sometimes it's a cofactor, sometimes it's a coenzyme. What's the difference? Cofactors are inorganic metal ions like iron and zinc. Coenzymes are organic, meaning that they're made out of hydrocarbons. Now, what about this? Enzymes can be blocked. They can be blocked pretty easily. Okay, so this is interesting. Remember, this is our enzyme, and then it has an active site. Now, I want you to learn this as well. This is called the allosteric site. It's another site that's not the active site. And it's usually in a different location. Well, of course it's in a different location, but it's usually pretty far. All right, so this is the anatomy of a regular enzyme. Okay, let's say this is the substrate. See how it's perfectly shaped to fit here. Okay, now if an enzyme is blocked, there's only two ways it could be blocked. It can either be blocked by a competitive inhibitor, which is a molecule that has a similar shape and it just sits in the active site, thus blocking the substrate. And that's not good. This is called a competitive inhibitor because it's kind of competing for the active spot. Now, a non-competitive inhibitor is something that can bind to the allosteric site over here. And when it does so, it changes the shape. Okay, so we just changed the shape of this from pretty much almost a perfect square with a weird little thing hanging out, right? But it's generally a square to a triangle. So now the substrate cannot bind here either. That's a problem. Okay, so there's two methods of enzyme inhibition: competitive, which blocks the active site, and non-competitive, which binds to the allosteric site, changing the shape of the active site. This is not always a bad thing. There are many medications that are enzyme inhibitors. For example, ibuprofen. Ibuprofen is a very useful and helpful anti-inflammatory medication. And why? It inhibits the enzyme involved in our inflammatory response. We have tons of pain signals. One of them is an inflammatory response, and a pain signal is called, one of them is called prostaglandins. Prostaglandins are a pain signal. Okay, so if we're blocking the production of prostaglandins, we're blocking pain signals. That's ibuprofen. Done. It's actually fascinating if you think about it. This is how we make the medications that help us get through hard times, like antibiotics. There are certain antibiotics that can inhibit the synthesis of I'm sorry, not the synthesis. That's a different type of antibiotic. This is a different, my bad. All right, so we have antibiotics that can block bacterial enzymes. Now, bacteria are these little tiny creatures. They don't have a lot going on. They don't even have organelles. We only have like 10 different lines of attack to kill bacteria and protect our bodies. Okay, this is one of them. Okay, we can block enzymes that are bacterial and thus killing the bacteria. There's also HIV drugs now that target the HIV viral enzymes. We have cancer drugs that inhibit enzymes that are involved in cell division. Okay, and that's important. We're going to talk a lot about cell division in upcoming chapters. All right, so this is the end of chapter 5. The last thing we're going to do in this chapter is watch a video about enzymes and watch the video. It is so important for you guys to watch the video and not just hear me talk the whole time. So watch the videos, all of them. I promise you they will help you in more than you realize. All right.
Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much, much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least to us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site, where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions. Reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. Now, with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So, as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. Some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts, and when they touch the Pac-Man, it makes this sound. It's like no, 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 no. And the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around, but enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production, or the lack of it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.